corner.
I know.
Yeah. Hi, I'm going to talk about mass spectrometry-based proteomics in my talk. In this talk, we will talk about like what is mass spectrometry-based proteomics and how it works, and how proteins are identified and quantified through various techniques of mass spectrometry-based proteomics, including discovery proteomics and targeted proteomics. At the end, we will briefly introduce how post-translational modification works using mass spec-based proteomics. So before we go into the mass spectrometry-based proteomics, we also want to understand what is proteomics. So proteomics is basically a concept. It's a protein equivalent of a genome, which appeared around 1994. While work with proteins, especially individual proteins, was began much earlier, like 50s or 70s, mainly by the electrophoresis. So the proteomics is really the study of proteins expressed by a biological system. Like you will study like what they are and how much they are, where they are located, and do they have modifications on them? What are their functions and activities? Do they have interactions between proteins? And there are many techniques you can use to study proteomics, like a mass spec, NMR, and some microscopy and the spectroscopy techniques. But today we are going to mainly discuss the mass spectrometry approach for proteomics. So mass spectrometry, which is even longer technique, which is measuring the intensity over the mass to charge ratio. So you will get a mass spectra of M over Z, which is a mass to charge ratio. And by doing that with proteins, you can either analyze the mass to charge ratio of protein itself, which we call it top-down proteomics. Or you can chop the proteins into smaller pieces, which makes actually easier for mass spec to analyze. So you will call this type of analysis bottom-up proteomics, when you analyze the peptides instead of the proteins. So today we are going to mainly, I will be mainly focusing on like a bottom-up proteomics. So the mass spectrometry-based proteomics was made possible, especially by the gene sequencing techniques, because after you have the gene sequence, you know the protein sequence, and then you can match those into what mass spectrometry present. Like you can use you can match those to the mass spectrometry. So that's why when you check, if you check the publications of genomics and proteomics in PubMed, you will see like there will be a more rapid increase in the genomics than the proteomics, earlier than proteomics. Yeah. And also the mass spectrometry-based proteomics is made possible by ionization techniques. So you need to get the peptides of proteins ionized so that you can send them into mass spectrometry. There are basically two types of mass ionization techniques, and both of them were recognized by the 2002 Nobel Prize in Chemistry. So the first one on the left is the matrix-assisted laser desorption ionization. So basically the matrix absorb the high energy from the laser and make charged particles. And the charged particle contains peptides or proteins, and also with lots of charges. And as the charged particle gets smaller and the matrix evaporates, in the end, you will left with only the charged proteins or peptides, and that you can send into the mass spectrometer. Similarly, but not with the exact technique, you can use electrospray, where at the end of the liquid flow, you can have a high voltage applied. And the high voltage will generate a lot of charged droplets too. And the charged droplets contained like peptides or proteins in water and with lots of charges, and the water evaporates and the charged droplet gets smaller, and you make lots of peptide or protein ions, and those can be sent into the mass spec. Because the electron spray ionization was using a liquid form, so it is very easy to connect to a liquid chromatography, which you can use for high throughput, large scale analysis of peptides or proteins. So this is actually more widely used in proteomics, while the MODI is mostly used in imaging. So today we are going to focus mainly on the electron spray ionization as the interface for mass spectrometry. And next, we will look at how the mass spectrometry-based proteomics works. What are the typical workflow? So basically, you have the cell of some samples like blood. You can do that too. Basically, we need to get the proteins extracted from the cell using cell lysis methods, and then digest the proteins using enzymes into smaller peptides. And then you can send the peptides into the liquid chromatography, so they separate the peptides into groups of peptides. And you can analyze those peptide groups and analyze further by the mass spectrometry. And later you will have like a data analysis tools to analyze those mass spectrometer and mass spectrum and match those two peptides. And for the cell lysis, because a cell always have like a plasma membrane, which is a protein lipid bilayer, the membrane needs to be disrupted so that proteins can be extracted. There are multiple ways you can do that if you search the literature. And you can use like organic solvents and bead beating, homogenizing, and you can even use detergent. So the techniques you choose must take into account of the compatibility with amount of the materials you are going to be processed, and what has the downstream applications you are going to use. So it's for the synthetic biological work of bacteria and yeast, the hardest to break is the yeast, and gram-negative bacteria are relatively easy compared to gram-positive bacteria. So after you have the proteins extracted, you need to use some proteases to digest proteins into peptides. Basically, the proteases will break the peptide bond between two adjacent amino acids. There are multiple enzymes you can use, and the most widely used are trypsins. You can see like the percentage of raw files which are using trypsins in this graph. And there are also some other like enzymes you can use. So different enzymes will cleave at different sites along the amino acid backbone in the protein. And also sometimes if you want to really get very high specificity, you can even combine two like enzymes together to make it more specific. And a lot of the times in our proteomics workflow, cysteine amino acid, because it is often get a double bond between sulfur and sulfur in the protein three-dimensional structure. Normally you need to reduce it, so you have easier mass spectrometer to analyze later. And after you reduce, because it is easy to react, so you need to alkylate it before you do the digestion. So in the end, you will have a cysteine amino acid alkylated in your data. And also, you need to, before you send your peptides into mass spec, you need to clean up any salts or detergents. because like salts and detergent are not good for mass spec. Now you can send your peptides into mass spec. So first, but before that, you need to use liquid chromatography to separate them. The reason, like in a system, even the simple like bacteria system, you can have like thousands of proteins, and if you digest them, you will make tens of thousands of peptides. So you cannot like just ionize them all together at once, because they don't get ionized together. So you need to separate them into liquid using liquid chromatography into smaller groups, so you can ionize them better and analyze them better. So using liquid chromatography, you first load the peptides onto the column. At the beginning, they start as a pack, so they are tightly together, and then because the column is packed with small particles, and small particles normally has like a C18, which is an octet diacyl, the peptide will interact with the C18, and it will take longer time for the hydrophobic ones to get out, and take less time for the hydrophilic ones to get out. So that in that way, you separate them by hydrophobicity. So in the end, you will have like peptide groups separated. And when they root, you will see like a nice separations between groups. And then you can send them through the electron spray. So at the end of the elution, you apply high voltage, and they make lots of droplets containing multiple peptides, but the small droplets will get smaller. So eventually they will contain maybe just one peptide in the droplet, and the water evaporates. So in the end, you will only have like a peptide molecular ions, which are sent into the mass spec. So there are two ways you can analyze those peptides. One is called the discovery proteomics, and the other is called the targeted proteomics. The discovery, so there are many differences between them, like they use different instruments, and the data acquisition and analysis are quite different too. So we will spend lots of time talking about these two methods today. So in the big picture, the discovery proteomics is analyzing the whole proteome. So whatever you get extracted and digested into the peptide form, you are going to analyze all. And target proteomics is mainly focusing on a small subset of the proteins of interest. For example, in metabolic engineering, you are interested in some metabolic pathways, enzymes. So you will be only analyzing those proteins. And also, because the way the instrument is running, discovery proteomics is mostly identifying and quantifying high to moderate abundance peptide proteins. And target proteomics has a better sensitivity. You can use it to monitor lots of the low abundance proteins. Sometimes, like even the peptides, you cannot see it in discovery proteomics data. You can still see it in targeted proteomics mode. So next, we are going to go to each different types of proteomics. So for discovery proteomics, there are multiple ways, multiple instruments you can use. The most popular one is the Orbitrap mass spectrometers, which we will use it as our example. So when peptides get eluted from the liquid chromatography, they are ionized and sent into the mass spectrometers. Okay, and inside mass spectrometer, they went through a series of lenses and analyzed by the mass analyzer. In this example, an Orbitrap is a mass analyzer. So remember that there are a bunch of peptides in this pack of a liquid chromatography elution. So that's why you will get lots of molecular peptide ions in the MS1 spectra. And so we call this spectra MS1. Yeah, it's molecular ion spectra of peptides. And once you have the MS1 spectra, you can further select certain molecular ions in the spectra. For example, if you select this pink one, and then you send it to a Clean cell, which in this case is called ACD cell, and you fragment it and fragment the molecular ions. You get lots of fragment ions, and you send them again to the mass spec analyzer. And so in the end, you will have a tandem mass spec spectra. So it is MS2 spectra. So you get lots of fragmentations from the peptides. So this is how we acquire the data in discovery proteomics, and how now we are going to go through like how we analyze the data. So basically you come to the MS2 spectra, and because the MS2 spectra is basically breaking the peptide bond again, if you have the ions, fragment ions from the C-terminal end, it is called Y ions. If it's from the N-terminal end, it is called B ions. And so you have lots of ions, and you can match those into the database. So you get the identification of peptides. So you know this molecular ion is from this peptide. And then you can go back to the intensity of MS1 spectra over time. So and then you get a peak, and you can use this peak to use as quantification for the peptide intensity. So in a real discovery proteomics data, you will have multiple samples, and you run each sample through the mass spec, and you get peptide intensity, and then you can compare the peptide intensity between samples. However, it's not possible for you to compare intensity between peptides, even in the same sample, because the different peptides will have different ionization efficiency. So the intensity, higher intensity, may not really mean it has high abundance in the sample. It sometimes may mean it has high response in the mass spec. So just to remember, like when you compare data between samples, you can compare peptides. Yeah, but you cannot compare between peptides, the intensity, or between proteins. So those, the ones I just introduced is called label-free quantification. So we don't have any labeling done in the sample preparation. Another way which you can actually get a better accuracy in quantification is to label your peptides. So after you digest your peptides, you can label them with different tags. There are multiple companies setting those tags. And so, for example, if you're using iTrack, you have 4 samples, you can label them with different mass to charge ratios of the tag. And you can combine those samples together since they are already tagged with different mass, and then send them to the mass spec together. So instead of looking for the MS1 intensity over time as your quantification, now you are looking at a pack of these tags as your quantification. For example, if you see the tags 140, 114, which has a low abundance, so that means that sample has a low abundance, and if 115 has a high abundance, then that sample has a high abundance for this peptide. So you still need to identify your peptide. So match your mass, like MS2 spectra to a peptide identification. And then you can see how the intensities are differentiating between samples for this peptide. So this is basically how you can do labeled method. So to compare the label-free and labeled method, the label-free still has larger dynamic range and the more depth of identification. Of course, because you are analyzing each individual samples separately, you will have more instrument time to need it. But you don't do tagging, so you have less preparation time. However, the label-free have more accurate identifications, and they use less instrument time. And but you need to have more preparation set up beforehand. But you can choose between those two methods for discovery mode. Now we are going to talk about targeted proteomics. So in targeted proteomics, there are many two ways you can do it. So one, using two types of instrument. So the first one is called selected reaction monitoring, which is using a triple quadruple. So somebody in the chat box was asking about what is difference between the triple quadruple and the Orbitrap. So basically, triple quadruple has 3 quadruples. And the quadruples are good for selecting mass to charge ratios. So in triple quadruple, you can do 2 selections. The first one, Q1, can be used for selection. Q3 can also use for selection. The middle quadruple is mainly for fragmentation. And for parallel reaction monitoring, you can use Q1, which is a quadruple again, to select precursor ions or molecular ions of peptides. And Q2, you can do like collision, fragmentation. And in the end, when instead of using a Q3, they use the Orbitrap or TOF. You can do a full like fragmentation analysis. And then when you do the data analysis, you can select just a couple of them to similar to what you will use in SRM. So you can use that just several of them as a quantification. So to compare, both techniques have good specificity, sensitivity, and reproducibility. But because the PRM is actually scanning lots of fragments, so the running time is longer for each peptide, precursor ions or molecular ions. So the multiplexing capability, so how you whether you can multiply multiple peptides into one analysis, maybe in that case, the SRM still is better. So today we are going to mainly focusing on like SRM-based approach for targeted proteomics. So in SRM-based targeted proteomics, again, you will have like a liquid chromatography elution of peptides, and you send those into mass spectrometer. And as I mentioned, it first come to the Q1, which is a quadruple, and you can select peptide of interest, and then send those into Q2, which is a fragmentation cell, collision cell. So you get lots of fragmented ions. And then you send those into Q3, and Q3, you can still do selection of one of the fragment ions. And in the end, you send to the electron multiplier for detection. So in this way, at the end, you will have a mass spectrum. You will have intensity over time for this particular molecular ion and the fragment ion pair. And normally in the SRM-based targeted proteomics, you not only just monitor one transition, one pair for each peptide. You normally monitor 3 pairs, so 3 different fragments for the peptides. And normally, like you need to have like isotope labeled internal standard peptides, which is to use isotope K and R, like a lysine and arginine at the C terminal instead of the regular ones. So you have a mass difference between the endogenous ones and the internal standard, but chemical and the physical properties of internal standard and the endogenous peptide are the same. So basically they will elute at the same time and they will have the same fragmentation patterns. And you spec those heavy peptides into the system. And so you have a mixture of endogenous and internal standards. And you send them together into the triple quadruple mass spectrometer, which is for targeted. And then you screen all the transitions, all the molecular ion and the fragment ion pair. And then you get the intensity over time. And you can use the ratio between the peaks and the curve for the endogenous peptides versus the peak and the curve for the internal standard peptides as the quantification in this case. So SRM is mainly for quantification, and you can even do absolute quantification if you are really rigorous and want to use pure peptides, which are normally more expensive than the crude peptides. So pure peptide means like we know exactly how much concentration of these internal standard peptides. So you can go, when you go back to the ratio, you can know exactly how much concentration of these endogenous peptides in the system. So if you use just crude, which are cheap and easy, you can still do like a comparison between samples, but you will not know like the absolute values of the concentration of the peptides you are interested. So there are so now we are going to discuss a little bit like how we do like post-translational modification using mass spectrometry-based proteomics. And post-translational modification, there are many types of them, which can happen in the system as listed here. And they can also happen at different levels at various species. So the most widely present post-translational modification is the PTMs. So a lot of people are actually studying a lot about phosphorylation. So a lot of people are actually studying a lot about phosphorylation. And we are going to use this as an example later in the next slide, how we can study phosphoproteomics. So because the low abundance of post-translational modifications, especially relative to the non-modified versions. So if you just run like a typical proteomics, and the non-modified version will overshadow the modified version. So it's really, really hard to see any post-translational modification if you don't do any enrichment. So if you really want to do post-translational modification, you need to enrich those peptides out of the non-modified ones. So you can specifically target those phosphoprotein, for example, phosphoproteomics. And in this case, for the phosphor, and there are also many other like things you need to consider. For example, the phosphoproteomics, because they are easy to attach to metal. So in your system, you need to avoid any metal contact with the like the columns or electrode spray. Also, like if you want to really see the fragmentations, especially the localization of the phosphosite, you need to use softer fragmentation techniques when you set up your mass spectrometry runs. And in the end, when you analyze your data, you also need to have some sophisticated softwares to help you to localize the phosphosites. So in summary, we talk about what is mass spectrometry-based proteomics, especially bottom-up proteomics, and also how what is the typical workflow. And we went into some details of about data acquisition and the data analysis of discovery and the targeted proteomics. And we also introduced some post-translational modification works with mass spectrometry-based proteomics. So in the next session, we will go even deeper into more details about this discovery and the targeted proteomics. Okay, so that's the end of my presentation. I can stop share.
Yuqian, I can help with a few of the questions. One of the last ones we got that you could address is, what would you do if the isotopically labeled peptide you wanted to measure using SRM or MRM does not exist? So is that ever a problem?
No.
Yeah.
So
because you've even done
this.
phospho ones too, right?
Yeah, so they can even make like PTMs, like for post-translational modifications on the peptides. So it's normally like all the peptides, if you give the company a sequence, they can sequence it, they can make it. The only problem with making a peptide is if it is too long. They have problem making very long peptides, yeah. But other than that, you can, synthesize most of the peptides.
Mm-hmm. And since arginines and lysines are so abundant in the peptides anyway, that it is probably most everything is following into the range that you want to see. And then you can tell the company what isotopic peptides you want to synthesize based on what ones you know will show up well in mass spectrometry too, correct?
Yes.
How many and how many peptides do you usually target for each protein that you're looking at? Is it
ideally, if you have two good ones, which are correlating well, that's the ideal case. Yeah, at least two. But you can target more if you have the budget, but normally two is enough.
Okay. And then you described TMT, but it looks like there's also a question about iTrack. So how do you label iTrack for quantitation?
I think in my example is with iTrack, but they are actually quite similar, like just different tag. So the small molecules are different. Yeah.
Okay, here's a good one. So it's always, formalin fixed paraffin embedded tissues. You know, that a lot of people that look at, biospecimens that are banked. So here's a question: if I have a FFPE sample and they're over 10 years old, can I still do a proteomic study with it?
Yes, I think absolutely. Actually, I already done those. I use targeted proteomics to do quantification of peptides with FFPE samples. You can absolutely do it. Yeah. Okay. There are protocols available. You can extract proteins, and a lot of the peptides are still relevant in the analysis. You can still see them.
And then here's another one about comparing abundances for discovery proteomics. You mentioned that proteins can be compared between samples, but peptides within a sample cannot be compared. And yeah, this is a good one that is always important to address. So I guess, do you want to talk about different ionization efficiencies for different peptides?
Yes, so peptides definitely have different ionization efficiencies. So to get a peptide to ionize well, some of the hydrophobic ones will ionize well because they will sit at the outside layer of the droplet, so they ionize well. So because different peptides have different ionization efficiency, so it's very hard to compare the intensities from mass spec and to relate that back to the actual abundance in the sample.
Mm-hmm. But if you typically you're gonna, if you see 10 peptides from sample A and 10 peptides from sample B, those are often the same peptides and they're going to have the same ionization efficiency. So you have a great way to do the relative abundance, since relative means each one of those peptides has the same behavior. It's a relative measurement of that same peptide across two samples. But yeah, and there's a lot of questions about, absolute abundances before you started discussing your targeted measurements. So that's,
okay.
Yeah, that,
Let me go back.
But
yeah, you can really do absolute with the targeted approach when you have the pure peptides. You may still have some variations, like especially also like in the peeled ones, when you order them, when you purchase them, they may say it's like 20% accuracy. So you may still have some variations, but it's maybe a lot of time it's good enough to do absolute quantification with that much variation.
Yeah. Here's a good one. How to select one or more than one peak to do MS again? Is it based on what's known? So I'm assuming that's if you're choosing a peptide, maybe for the, if I'm interpreting that correct, maybe for the targeted analysis, how do you choose what peptides you use to make isotopically heavy versions of?
Oh, yes, we will talk about that in the next presentation. But basically, you are trying to choose a good flyer, which can fly well in the mass spec. So you have good intensity. The higher the intensity, the more sensitivity you will get from your endogenous detection or quantification. And also, you need to look for some peptides without any modifications. So you are looking for the peptides which can be a true representation of the protein abundance. So there are also some other things to consider too, to find the good peptides.
Okay. Maybe, how are we doing on time, Leanne?
We have a couple minutes. We're scheduled to break at 10.
Okay.
So you have a few more minutes for questions.
See, I guess one that I already wrote a response to, but someone asked a really good question about using MALDI, so matrix-assisted laser absorption ionization, for spatial profiling of peptides across the surface. So if you have a microbe that's growing on a plate, or you put that on the plate. So in terms of peptide imaging, if there's endogenous peptides you want to look at, that's a very powerful technique for imaging it. But one thing that, all these peptides are fighting for charge. So if you're, ablating it directly from there, you can get ion suppression. And that's, Yuqian did a really good job talking about all the liquid chromatography and other things we do to try to separate peptides from each other, so you can get really nice measurements from individual analytes. So do you want to do on surface or in depth? So it always depends on the biological question of interest. Okay, so someone has a question about spectral count versus relative abundance.
Mhm.
I guess, do you want
Spectral
to,
count.
yeah, maybe describe what a spectral count is versus a relative abundance?
Yeah, spectral count is like count how many spectrum of each peptide you have in the sample. So you see in the sample. It's really not a good quantification, even for the same peptide across samples, because like when you run the instrument, you are not constantly like fragment this peptide always. So because the way we run the discovery mode is like we just select the maybe top 10 from the MS1 spectra to fragment. And sometimes we even skip for a certain period of time. So we can get as much like a molecular ions to get fragment as, yeah. So in that case, you can randomly missing some spectrum for this, for example, for this peptide. Yeah, so it's not really a good comparison between, yeah, to quantify peptides.
Yeah, you get really relative versus getting that nice
Yeah, like
abundance peak.
The spectral counts can be, yeah, random. Like if you run the same sample twice, you may not get the same spectral count for the same peptide.
And then there's a question about, is it known how much post-translational modifications like phosphorylation affect ionization efficiency of peptides?
If you're thinking of like ionization, like phosphor-related peptides, normally is like more hydrophilic. So if you have some competing peptides which are more hydrophobic, the hydrophobic ones will be at the outside of the droplet, and the hydrophilic ones, which is phosphorylated ones, will be at the inside. So it will be easier for the hydrophobic ones to get ionized and then the hydrophilic ones. So phosphopeptides really performs less, so ionize the less compared to the non-phosphorylated ones. Yeah, so they definitely have some effect.
Great. And that's kind of going back to not all peptides are equal.
No.
But we account for that. We know that, and it goes into the analysis. I think we're at a good point where, you know, that we can address the other questions in the chats.
Yeah, we have a half-hour break from 10 to 10:30, and maybe we can keep looking at the questions in Discord. I think questions are also starting to probe at how we analyze the data, which is what we'll start to talk about at 10:30. So that's awesome.
And that's, you know, as we stepwise through these presentations, you know, you're getting more and more in-depth knowledge. So by the time everyone walks away today, I think they'll have a pretty good idea of what all the terms are, and we should be in good shape.
Awesome. Thanks so much, Kristen, Yuqian. Those were awesome presentations.